There are many questions surrounding this idea of junk DNA. Is it really junk? And if it is, why there is so much of it in our genomes? Does it have a function? What does evolution have to do with it? And what does it have to do with evolution? And maybe most importantly, does it have anything to do with dysfunction and perhaps finding cures? So today I come across the street to the Salk Institute to meet up with a couple of friends. The Salk Institute's own Rusty Gage and UC San Diego's Miles Wilkinson. Rusty, Miles, and I will grapple with those questions and bring up more that are sure to arise as we dig deeper into the world of junk DNA. Miles, I'll start with you. What is about junk DNA? I mean, how come our cells have so much extra DNA material inside them?
Well, I think that actually we need to determine if that's actually true. We need to know what proportion of the genome actually is junk. Mm-hmm. So by junk, I suppose the definition for that would be DNA that is of no selective value to the organism that has it. If that's how we're defining junk, I think the big question is what proportion of the genome actually is junk.
Right.
So there's been a kind of a swing of the pendulum with regard to this. So especially in the 1980s, there was quite a bit of thought that most of the DNA was junk. And part of that came from the fact that coding portion of the genome, the portion that actually encodes proteins, only represented an extremely small proportion of the genome, something like, back then it was thought to be maybe less than 5%. We now know it's actually only about 1% of the human genome. And so what did the rest of all this DNA do? And at that time, we didn't really know much about other things that DNA could be doing. There was a lot of speculation involved in chromosomal structure, et cetera. But the idea was proposed that a lot of this could be what's called selfish DNA.
Right.
This is a term that was proposed by Richard Dawkins, who had the famous book called Selfish Gene. And the idea is that, well, this is kind of a stark view, but that cells or organisms are really just a place for DNA to replicate itself. Mm-hmm. It was just a host. And what's really going on is the DNA is just finding ways to make more copies of itself. And the organism is sort of secondary. Obviously, that's not completely true, but it's one way of looking at things. Mm-hmm. And if basic idea with selfish DNA is if DNA has the ability, a piece of DNA to replicate itself, such as what we'll talk about perhaps later, transposable elements, where they can leave a copy behind, they don't need to provide any benefit to the host. They have an ability to proliferate. And so the idea came out that, well, maybe most of this DNA is really not doing anything for the host. Mm-hmm. So that was sort of the 80s, and I think that depending on everybody had a different point of view, but a lot of people tended to think most of the DNA was junk. And then we discovered after the year 2000 that most of the genome is transcribed, meaning the DNA is made into a transient form called RNA, which ultimately encodes protein. And 80% or something of the genome is transcribed. And so then the kind of pendulum swung, and lots of people said, well, if it's transcribed, it must be doing something. Mm-hmm. I personally don't take that point of view. I think that it takes work to prevent transcription, and you can have low-level transcription that's irrelevant, just because it's actually, once you have transcriptional machinery, you're going to have all kinds of transcription occurring. And a key point is that majority of this transcription in the genome is below 1 copy per cell. That means there's less than 1 copy of the RNA per cell. So it's, in my mind, hard to imagine that that is going to be functionally important. It's possible, certainly, but so I think the big question is where in this continuum, how much of the DNA is junk? And a problem with the term junk is that it suggests that that is what something is, and we've defined it, and we're done. Mm-hmm. The problem is, as research continues, we're going to discover that more and more pieces of DNA actually do have a function. And so, we have to think of perhaps a good term that's a little more open. There's a lot of reasons to be positive and think that as time goes on, we'll discover more and more functions for what we call the non-coding DNA. The coding DNA, I think everybody agrees, is generally functional, not necessarily all of it, but most of it. But this non-coding part of the genome, which is in the humans 99% of the genome. And I'll just say one last thing in terms of the idea that yes, we will discover functions clearly for a lot of these things, but what percentage of these things are we going to find functions for? Well, now there's I just looked this up. There is a plant called a bladderwurst, and I guess it lives in very wet sort of areas, and it has 97% of its genome is coding. Only 3% is non-coding. So that says that in a simpler organism that's presumably simpler than us, that you don't need much non-coding DNA. We have 99% non-coding, they have 3%. Now, you can argue that's because we're so advanced, we need the non-coding DNA. Okay, fair enough. But then, if you look at, say, salamanders and lilies, two very different kinds of organisms, they both have about 10 to 20 times more DNA than we do. And I think most people would argue that they're not more advanced than us. Lily's a nice flower, salamander's an interesting reptile. They have, so, I think you really have to question, how much of this non-coding RNA is really going to be, in the end, functional. I'm certain a lot of it will be, and it's going to be interesting to find out.
And I'm not expecting us to solve this issue now, but I'm turning to Rusty now, trying to explain, how come that this much of DNA accumulates in the cells? And part of that is through this mechanism of jumping genes or retrotransposons. Can you explain, Rusty, how the cells accumulate DNA over time?
Well, let's go back to single-cell organisms during evolution where they had a very great way of protecting their genome. It was these gene editing systems that are now popular, called CRISPR-Cas9, for example, that we use experimentally to cleave out parts of the genome or put in new pieces. Well, single-cell bacteria that had these kinds of systems used that to protect themselves from invaders. So they kept their genomes pretty tight, and at one point in evolution, these single-cell organisms changed and the editing systems were mutated. And once they were mutated, the genome started expanding because the mobility, either viruses coming in or making copies of themselves expanded. And that was, some people think of as the beginning of the expansion of the genome. And it was in part because there was no editing system to prevent it. Fast forward to mammalian systems, and we still have these mobile elements that are moving around in the genome. And we've actually, or mammals and plants, have developed a lot of mechanisms to control this mobility, and most often to shut it down, but not always, and also in very, very programmed ways, so that it allows for mobility to occur at certain times during development, for example, and then it's shut down very tightly. So we have these elements that exist within our genome, and in fact, it's estimated the human genome now that something close to 50% of the genome is made up of the residuals of these mobile elements that started a long time ago. They're not all still active now by any stretch of the imagination, but there are elements that are still active. But some of the elements that were active in early species or early parts of evolution are still hanging around. They've been mutated such that they're not able to move anymore, but their genome sequence has been adapted or co-adapted for other purposes. And there are examples of what are called human endogenous retroviruses, is a HERV they're called, that were active early on in evolution, became silent, and yet when we began to map out in the genome parts of the DNA which are involved in regulating the expression of this protein coding sequences that Miles was talking about. Turns out that these pieces of these HERVs are often intimately involved with regulation. They're called enhancers. They have elements of enhancers within them. And so one imagines, and stories are emerging now where a HERV jumped into a location at some point during primate evolution and played a role in regulating the expression of a gene that was downstream. Now, because these HERVs are like mobile elements, they have a lot of repetitive sequences in them, and that makes them a little more vulnerable to mutation. A HERV could have jumped in at a species before it split, for example, before we split from chimp and human, and it's carried in both, but then a subsequent mutation in that HERV would have changed the expression of this gene downstream in both cases. So there's a case where we have mobile elements perhaps falling in the range of junk, playing a role, by chance, by where they landed, played a role in gene expression, but then by virtue of the features of the DNA, they are subsequently involved in the evolution of the expression of proteins.
Right.
So while I think it's true that there is probably lots of DNA for which we don't understand its function currently, this is a great time in biology for looking at an evolutionary perspective and finding out when events occurred and what role they may have played early in evolution, and that subsequently they may be in fact non-functioning now, but they had a function at some point and now they have less of a function, or vice versa, where they had no function previously and by virtue of where they are now, or some mutation that occurred and they have function. Overall, there are two driving forces in evolution. One is we think of selection. We always put the pressure on selection. But the other feature is diversity. Mm-hmm. In order for a species to persist, there needs to be, it's an evolutionary process by which diversity occurs and then selection occurs on that to adapt to whatever that environment or that organism living in. And perhaps the mechanisms that are generating, or the junk DNA itself, is a template on which diversity can act without disturbing the ongoing processes. So you need this flexibility, you need this variability in the genome on which to select based on the environmental circumstances that you have for that time, for that species.
Mm-hmm. I think you gave awesome examples of how this mobilization of those elements provide a fertile soil for evolution to act. And this is becoming more and more clear with more examples like that. But you and I, we go back to the early 2000s where we figured out that one of those retrotransposons are highly active in the brain. And so was that a big surprise for you to realize that these are actually active, still active in individuals?
Yes.
For me too.
It was a great time for us during the realization that this was happening. I think from a personal perspective, I have been working and had an interest in retroviruses per se as tools for gene delivery. Mm-hmm. And so the features and the structure of the retrovirus was fertile ground for investigation for me. I think I knew less and paid less attention to the line elements per se at the time, but when you look at the structure and see that they have a lot of these common features, I think that was one of the attractive parts. I felt comfortable in that DNA sequence. That they were active in early neural progenitor cells, or just before they became neurons, was a surprise, not just for us, but for the whole community, because the only place where it was thought to occur was in germ cells previously. So we met with a certain amount of resistance until others were able to replicate it using analogous tools and things like that. But it's still, I think there's been a focus now, the realization that the features of these particular mobile elements requires that the nuclear membrane is broken down for them to get back into the DNA and insert themselves. And there are more and more studies showing how they may play a role in cancers in particular. But I think that's been going on since Hay Kazazian's first studies with line elements in cancer. Mm-hmm. I think the interesting line now is what role they might be playing in natural behavior or in development, during development, what role they play in generating diversity subsequently in behavioral changes that occur.
Mm-hmm. And while somatic neuroprogenitor cells might be loose in the control of those jumping genes, the germ cells are quite strict about their mobilization, right? Mm. So, Miles, you also work on factors that inhibit the activity of these retrotransposons. So what happens if you don't have those factors in the germ lines?
Could I also respond to what Rusty was talking about? And then I'll get to the germ line in a minute. Okay. So I agree that selection, is a key tenet of evolutionary biology. But in the 80s, there was a kind of a shift in thinking that selection doesn't always need to operate on individuals in a species.
Right.
It can also operate within a cell.
Absolutely.
And so, that this gets now back to these pieces of DNA that are able to replicate themselves. Mm-hmm. And basically, you have competition between different replicators in the genome, and whoever can replicate more can win out, right? And so, in your case, these jumping line elements that I guess are jumping at increased frequency in neural progenitors, I think is very exciting. I agree, but I guess the question is that because it is doing something functionally important, or is that because neural cells are allowing for it? Because the key thing with jumping genes is, we'll talk about germline in a minute, mm-hmm. but the germline is critical because, these things are going to get passed on, and when they jump to a new spot, they're going to create mutations, which usually probably have no negative effect, but sometimes they will if they jump in the middle of a gene. But perhaps in neurons, just to take the devil's advocate mm-hmm. position, it doesn't bother them too much. There's not enough selection pressure against it, and so that's why it occurs. Now, for it to be functional, from my very naive point of view, it seems like it would be preferable if there was some selectivity to their jumping. Not absolute selectivity. You could still have diversity, as you said, but some kind of selectivity, because if it's completely random, it's hard for me to understand how that would be functionally useful for the organism.
Yeah, no, I think those are all good points. I think just to take up this point that you're raising with regards to randomness, right? I don't think this is a diversion because I think it's at the heart. It's the heart of the issue.
No, it's a fun topic because you published both of you published a lot of nice things on this area.
We think of randomness. When you think of randomness, you think that all options are open if something is fully random. But another way to think of genomic randomness is randomness with constraints. Mm-hmm. That means that there's certain features of randomness or certain features of an activity that limit its mm-hmm. full randomness. So, maybe for example,
less randomness
or controlled randomness. We know, for example, that the chromatin needs to be open. Mm-hmm. So if it's open, then it's likely to be active.
Right. So that reduces the number of target sites.
That reduces the targets significantly. There's a significant amount of work going on right now in the hemopoietic field about whether or not certain areas of the genome are more vulnerable to damage than other areas. Mm-hmm. And it may turn out that in addition to these structural constraints like open chromatin or other features like activity, mm-hmm. There may be even, features about the genome that reduce the number of targets that actually are there. So there may be more targeting. Yeah. This we'll have to see, but it is beginning to be reduced. So maybe to answer that will take another 10 years to see what is the relative thing. Mm-hmm. But another way to look at it is that when we talk about function, I would include within the domain of things that are function just generating diversity. So especially in a neuron, so if you have a neural progenitor cell that's not going to be carried onto the germline, that it's open for business, mobility can occur at a certain stage, it generates on a single cell basis, a mosaic basis, diversity within that cell. Mm-hmm. Given the fact that most cells, after they differentiate into a neuron, never divide again. So you have that same cell in your brain for the lifetime of that cell. You're not concerned so much about replication and addition, but what you've added is an element of diversity within each individual cell that adds that expanded capacity of the cell to function. And in a theoretical sense, this would also add to the individual differences that exist between people. So when you have things like identical twins with quote, exactly the same genome, they may not actually be exactly the same genome if there are these events that are happening that make them somewhat different later on. This is an argument, or this is a discussion about these sorts of things.
So Rusty, if it was beneficial to have this this mosaicism in there, or that's too fancy a term, but individual differences in different neurons with different elements jumping in different places in different neurons. Ultimately, for that to be of selective value, we would need to have more babies as a result of it, right? I mean, that's how selection works, right? If it's something that gets passed on, you are more likely to have children and pass on that ability. So do you see it as being somehow?
Yeah, I think it's not where they're jumping that makes that we're talking about. We're talking about the mechanism that allows the jumping to occur, and not necessarily specifying exactly where they go. So that there may be features like methylation or marks on the DNA that have some regulatory control about how much can happen in one cell or one individual. That could be passed on genetically. So, or some other genetic mutation where the enhancer on that element is not bound as tightly because of some mutation that is inherent in that family. So I don't think where they land has to be passed on, but the mechanism by which that allows them to move. But
I'm just frankly having trouble understanding how elements that are jumping somewhat randomly are going to somehow, over a long period of time, lead to, more likely to have children, because ultimately that's how genes take over, right?
Let me try to do an analogy with the immune system. And I know it's not perfect, but so the immune system provides all this level of diversity and variability and have the VDJ locus where this recombination happens, right? The selection is not at the level of the individual, the recombinations that you have and I don't, or the diversity of your immune system. But we are selecting for individuals who carry that mechanisms to be able to do that. If now we have individuals without that mechanism, they will probably not survive. So I know it's not the perfect analogy, but I think the lessons from the immune system might tell us that the pressure is on the mechanism. No?
And to that
has immediate value, because you basically providing T cell and B cell mediated immunity, and if you don't have that, you essentially have AIDS or worse, and you die. But I don't know what the negative effect of not having jumping genes in neurons.
I think you've really hit the nail on the head there, and that this is a new field of endeavor, and the deletion experiments are just currently underway. So that means what happens if you suppress line activity or delete that activity through some mechanistic studies? And I think those need to be done to address your question. But I think part of the problem there is there's so many active elements that it makes this experiment a little more difficult to do. And it's interesting to note that we often will then talk about it in terms of humans, but really we go to our animal experiments to do the experiment to see what happens. And whereas We believe, for example, that humans have about 150 active elements that are still relatively active in our genomes. The mouse has 3,000.
Right, I know. This is a case where humans would be a better subject.
Yeah.
Exactly. So, but I can tell you those experiments are underway.
Well, again, if there's selectivity to it, as you said,
Yeah.
you know, there's some constraints.
Exactly.
Then I could really see how it could be
Yeah.
of selective advantage. And
But
useful.
one thing that I think it's another argument on another piece of evidence is the fact that when you knock out or we remove the factors that controls retrotransposition or mobilization, you have a dramatic effect.
On development, yeah.
On development, yeah. So, and sometimes for neurological,
Right.
for the neuro part as well, neurological problems. So those animals in experimental models, they show neurological deficits.
Right.
So there is something there, but I agree with Rusty. I mean, these are, it's an early topic. We are taking advantage of these new tools in molecular biology to really manipulate the genome in a ways to answer those questions more precisely. But let's go back to the germ cells.
It's going to be very exciting. Actually, I'm
Yeah,
looking
it
forward
is.
to
It is.
finding
It
out.
is. It's great fun.
Yeah, and just mapping, for example, where those sites are, where
Exactly.
they're jumping,
Yeah.
is going to be, which is hard. I know it's very difficult.
And it might be different
Especially
brain
when you've
regions.
got-
Yeah.
Yes, of
Yeah.
course, different brain
Yeah.
regions. Yeah, going to the germline. Well, I think the way I look at it, and it may be very naive, but it's sort of a dynamic balance. So, on one hand, the transposable elements are jumping, and also not just jumping, but sometimes just being expressed, which can cause deleterious effects to the germline directly. These germ cells, they give rise to the next generation. As you said, there's evolutionary benefit to have the diversity caused by these things jumping, but there's really no selective value in that. I mean, evolution doesn't look into the future.
Exactly.
It just this is just raw material that is useful later on, but it's not purposeful.
Right.
And so, really, I think most of the effort has been in the germline to suppress the jumping. But it's not perfect, which is good because then that allows a little bit of jumping to provide a little bit of diversity. And so it's sort of a balance. And there's all kinds of mechanisms, including a large part of or a small part anyway, of the non-coding RNAs, piRNAs, as they're called, are clearly important in suppressing the jumping of some of these genes. Another thing is a chemical modification we're all familiar with that occurs on DNA, methylation.
Yep.
And that also suppresses the jumping of these elements. But there are 2 times during development when there's genome-wide demethylation. The methylation group is removed, both during gamete generation, but also in the early embryo. And I wouldn't say it's completely removed, but about 90%. It's very, very dramatic. And when you demethylate, you open up the DNA to be transcribed, including these often nasty, selfish DNA jumping genes, right? And so, but it's a balance. And so that's why there's a lot of mechanisms to try to suppress it.
Okay.
It's interesting. I was thinking about this, that some of the mechanisms that are used to regulate other parts of the genome likely emerged initially to suppress these mobile elements. And then they were exapted or taken, their function was taken. And methylation is a really good example of this, where methylation is used to suppress genome or other kinds of epigenetic marks are used to regulate gene expression and likely early on these mobile elements to silence or regulate their activity. And then this was exapted for other purposes.
And now we make use of it.
And now we make use of it.
That's right, that's right.
And it is interesting to go back to this, the early stages of the cells, right, when the DNA start to expand. But there is a point where they need to restrict that. And that's when these factors in the cell learn that probably is not advantageous to the cells to just let them grow. I don't know if I have any example of a mechanism where the genome is expanding, still expanding, in a geological time where we can actually see that. I don't know if there is any examples of that, because that would be an interesting species to study how this
To see how it adapts
how
to
they
all that
adapt.
extra
Yeah,
DNA.
yeah. I tend to think that this is all about energy. There is a time where the cell said, well, I cannot spend all my energy trying to replicate DNA. We need to restrict this.
Right.
And I mean, selection was to favor the elements that will control the jumping genes.
Yeah, there's a gentleman named John Maddock. I think he's still in Australia. I'm not sure.
I think so.
He's an Australian anyway. And and he shows this chart of complexity and versus non-coding RNA. And basically, as you go to more complex organisms, there's more non-coding RNA. And generally, I believe his theme has been that suggests that the non-coding RNA must be doing something really important for these higher organisms. But an alternative view is just that the higher organisms are able to put up with it more.
Right.
And then that kind of comes with what you're saying. So bacteria, depending on the species, divide every 20 minutes. It can't afford
Right.
a lot of unnecessary stuff, junk DNA. And so they have very streamlined genomes. But the way I think of bacteria as, species that never went to college. They have reduced options,
Right.
but they're also going to survive a nuclear warhead or something like that. At least some of them will. But they have less options because they don't have so much of this, what we'll call junk DNA, which, as Rusty and I have said, can generate so many things for the future. It may
Right.
not be useful
Right.
initially, but ultimately. Whereas we have, in my opinion, a lot of superfluous stuff that isn't doing anything, but it doesn't mean it won't be useful later.
Yeah.
So.
Yeah.
Was there like an example some time ago on the cockroach genome that is so big and so large that why it can be protected or so resistant to several environmental factors? I think there's a recent paper on
Yeah, I
that.
can't remember. I think you're right.
Yeah.
Right.
This is a cockroach.
Yeah.
Right, and so that may be true, but I guess the question is, was that selected for that purpose?
No,
Is that
exactly.
why?
Right.
Yeah.
Yeah, but there are these early hypotheses that the junk DNA is all about protection, right? I mean, it covers the important pieces of DNA, and once you have a mutation by sunlight, by whatever environmental factor, it goes to this part that can afford to be mutated.
That's right.
But not on the protein coding genes or the most relevant genes.
Yeah, in fact, investigators, I think even as way back in the 60s, way before my time, and in the 70s, including Ono, famous evolutionary biologist, predicted that you couldn't have more than about 30,000 genetic loci or pieces of important genetic information in the genome because the mutational load would be too high. And that's kind of held up, at least in terms of coding genes.
Right. You could.
Because it's nice to have all that extra DNA to hit. Because let's
To
face
absorb.
it, most mutations, even in coding regions, are probably neutral. I figured neutral's number one, number two's bad, and then occasionally it's a good mutation. It's actually helpful. But when you have millions and billions of years to work with, ultimately the good guys get selected.
Right. Okay, I think we are ending our discussion here. Thank you, Rusty. Thank you, Miles. I think this is a very helpful discussion.
Thanks.